Jnanam paramam dheyam. Knowledge is supreme.
Friends, in this lecture we are going to look at how to estimate or how to identify whether a particular catalytic reaction, particular heterogeneous reaction is diffusion limited or reaction limited. Often in industry several experiments can be performed, but it is important to understand under what situations, under what conditions the heterogeneous reaction will be diffusion limited or reaction limited. And it is very useful to have certain tools which can be used to identify these regimes. So, what we are going to see today is to look at how to detect from the experimental data, that is the observed reaction rates under different situations and different conditions, how to identify what is the under what situation the heterogeneous reaction is diffusion limited and under what situation it is surface reaction limited. So, the diffusion limited situation can actually be of two types. One can be the external diffusion, so diffusion limited experimental conditions can actually be diffusion limited reaction conditions can be either internal diffusion controlled or the external diffusion controlled. The other possibility is where it is the reaction limited, the conditions are reaction limited. So, as a first step we will look at the Vibratory criterion, which is often used in industry in order to identify whether the reaction is internal diffusion limiting. So, we will start with Vibratory criterion, and so the Vibratory criterion is actually used to answer this question: is internal diffusion controlling the internal diffusion limiting? Are the current conditions are such that its internal diffusion limiting the actual performance of the heterogeneous catalytic reaction? So, suppose if from experimental data we are able to obtain what is the observed reaction rate. So, if the observed reaction rate is given by, let us say, minus RA prime OBS is the, let us say it stands for the observed reaction rate, and we will use this nomenclature for the rest of the lecture. And let us consider a first order reaction where A, which is a reactant which undergoes a certain heterogeneous catalytic reaction and results in forming a product, and let us assume that it is a first order reaction. So, now for a first order reaction, in the last few lectures you would have noticed that there is a relationship between the effectiveness factor and the Thiele modulus. So, for a first order reaction, the relationship between the effectiveness and Thiele modulus is given by eta, which is the effectiveness factor, that is given by 3 divided by phi 1 square. 1 stands for the 1 here refers to the first order reaction, 3 by phi 1 square multiplied by phi 1 cot hyperbolic phi 1 minus 1. So, that is the relationship between the effectiveness factor and Thiele modulus for a first order reaction. Now, this equation can actually be modified as eta into phi 1 square, that is equal to 3 into phi 1 cot hyperbolic phi 1 minus 1. So, all I have done is just multiply the whole equation with phi 1 square. So, once we do this, we can now define a new parameter called the Vibratory parameter. So, we define a parameter called the Weisprater parameter. And this parameter is, you see CWP, C subscript WP represents the Weisprater parameter, and that is equal to eta into phi 1 square. Okay. So, we define a new parameter called CWP, which is equal to eta into phi 1 square, and that would be equal to 3 into phi 1 cot hyperbolic phi 1 minus 1. So, now what is this parameter? So, let us look at what is the meaning of this parameter CWP. So, we can look at the meaning of this parameter. So, CWP is eta into phi 1 square. The effectiveness factor is essentially the ratio of the observed reaction rate to the reaction rate evaluated at the bulk concentration CAS. So, if the, so that will be bulk observed reaction rate divided by the reaction rate at the bulk concentration. If we assume that there is no mass transfer limitations, that will be equal to the concentration at the surface of the catalyst, and that multiplied by the Thiele modulus. So, phi 1 square is nothing but the reaction rate evaluated at the surface concentration if there is no mass transfer limitations, divided by the diffusion rate of the species that is actually undergoing the reaction. So, that will be reaction rate at CAS divided by the diffusion rate of species A. So, now from here we can actually rewrite this expression. So, you can see that we can cancel off the denominator in the first expression and the numerator in the second expression, and we can rewrite this Weisprater parameter as observed, as a ratio of observed reaction rate divided by the diffusion rate of species A. So, this parameter, Weisprater parameter, provides it provides a mechanism to compare the observed reaction rate divided by the diffusion rate of the species A. So, let us look at what we can do with this Weisprater parameter. Remember that the objective is to find out whether the reaction is being conducted under internal diffusion limitations. So, now effectiveness factor eta, if we put the expressions corresponding to the effectiveness factor and the Thiele modulus into the Weisprater parameter, what we see is that effectiveness factor is the observed rate divided by the reaction rate at the surface concentration, and phi 1 square, which is the Thiele modulus, is given by the reaction rate per unit surface area of the catalyst multiplied by the surface area of the catalyst per gram of the catalyst multiplied by the density of the catalyst into r square, where r is the radius of the catalyst pellet in which the reaction is being conducted, divided by the diffusivity of the species multiplied by the concentration of the species at the surface. And so, that is equal to minus RAS into rho c into r square divided by CAS. So, now plugging in these two expressions into the Weisprater parameter, we will find that CWP is equal to eta phi 1 square, and that is equal to minus RA observed rate multiplied by the density of the catalyst rho c multiplied by r square, which is the square of the diameter of the pellet that is used in which the reaction is occurring, heterogeneous reaction is occurring, divided by the diffusivity divided by the concentration of the species at the surface of the catalyst. So, now if we observe this carefully, if we observe this expression carefully, we find that all the quantities which is represented here, they are either a measurable quantity or a known quantity. For example, they are all measurable or known quantities. All information present in this particular expression, they are all measurable or known quantities. So, the measurable quantity here is the reaction rate. So, the reaction rate is the measurable quantity in this expression here, and the density, radius and the diffusivity are again known quantities, and the reaction rate and the concentration on the surface are all measurable quantities. So, once we know these quantities, we know what the Vibrator parameter is. So, the Vibrator condition or the Vibrator criterion is that the Vibrator criterion is when the parameter CWP, which is the Vibrator parameter, if that is significantly smaller than 1. If that is significantly smaller than 1, which means that the ratio of the observed reaction rate to the rate of diffusion of the species into the catalyst, if that is much smaller than 1, then it means that there are no diffusional limitations. No diffusion limitation, internal diffusion limitations. Now, this also means that there is no concentration gradient within the catalyst pellet, because the diffusion is very fast, and so even before the reaction occurs, all of the catalyst is now replenished with the reactant species. So, therefore, there is no concentration gradient within the pellet, within the catalyst pellet. So, that is an important parameter. So, once we know these parameter, which can be estimated from the measurable and the known quantities, one can actually decipher whether the reaction is being conducted at the diffusional limitations or not. So, when Vibrator criteria parameter CWP, if that is significantly larger than 1, then this implies that there is a strong internal diffusion, limitations. Strong internal diffusion limitations, and in fact, it means that the strong internal diffusion limits the reaction severely. So, simply by using the observed quantities, that is measurable quantities, and some of the known properties of the system, one can actually decipher using the Vibrator criterion whether the internal diffusion is limiting the reaction or not. And that is an important aspect when it comes in practice. It is also important to note that while the Vibrator criteria can be used for non-first order reactions, the effectiveness factor in theory modulus for such reactions cannot be obtained using the method illustrated in this lecture. So, let us look at a specific example. So, let us consider a first order reaction A going to B. Let us consider a first order reaction. Now, an experiment has been performed with different catalysts, and let us say that there is experiment 1, in which the catalyst of 10 millimeter diameter was used. 10 millimeter diameter was used to conduct the heterogeneous catalytic reaction, and let us assume that it was performed in an appropriate reactor such that there is no external mass transport diffusion limitations, and so which means that the surfaces, the concentration of the species at the surface is equal to the concentration of the species in bulk, and the reaction rate that was observed is about 0.18 into 10 power minus 2 moles per gram catalyst per minute. That is the observed reaction rate for when the reaction was conducted using a catalyst pellet of 10 millimeter diameter. Now, suppose the same reaction was conducted under same conditions, except that a much smaller pellet was used. So, there is another experiment where the pellet that was used is 1 millimeter in diameter, 1 millimeter spherical particles, and the observed reaction rate is 0.9 into 10 power minus 2 moles per gram catalyst per minute. So, now because the experiments were conducted such that under appropriate conditions that there is no mass transfer resistance. So, here we need to estimate whether it is internal diffusionally controlled or not, whether the internal diffusion are there any internal diffusion limitations. So, that is the first question. Then the next question is find the effectiveness factor eta and the effectiveness factor and the Thiele modulus for both these cases, and what should be the radius of the pellet such that there is no diffusion limitations. So, the question is find the effectiveness factor and Thiele modulus and what should be the radius of the catalyst such that there is no diffusional limitations. Okay. So, how do we handle this question? So, we have just now learned the vibrator parameter. So, vibrator parameter is basically based on the observed quantities such as the reaction rate, etc. So, we can attempt to use the vibrator criteria in order to find out the effectiveness factor and the Thiele modulus from the observed reaction rate. So, the Thiele modulus effectiveness factor relationship suggests that eta into phi 1 square, which is the vibrator parameter, that is equal to 3 times phi 1 cot hyperbolic phi 1 minus 1. Now, so this is which is equal to the vibrator parameter CWP, and that is also equal to the observed reaction rate multiplied by the density of the catalyst multiplied by the square of the radius of the particle that is used divided by the diffusivity into the concentration at the surface, because the mass transport limitations are not there. So, let us say that the surface of the catalyst concentration of the species in the surface is equal to the concentration of the species in bulk. So, from here by looking at this relationship we find that minus R A into rho c into r square divided by diffusivity into CAS, that should be equal to 3 times phi 1 cot hyperbolic phi 1 minus 1. So, that brings a relationship between the observed reaction rate and the Thiele modulus. Now, for each of these runs, each of these experimental runs, we could actually find out what this relationship is, that is how the reaction rate at a given particular particle, radius of the pellet, how is that related to the corresponding Thiele modulus. So, for experiment 1, this is given by minus RA1, which is the observed reaction rate when the catalyst pellet is let us say 10 millimeters, multiplied by rho c into R1 square divided by dE into CAS, that should be equal to 3 into phi 11. Now, phi 11 corresponds to the Thiele modulus of the first order reaction for the first experimental run and multiplied by cot hyperbolic phi 11 minus 1. And similarly, for experiment 2, for the other radius of the pellet for which the experimental data is available. So, there will be minus A2 into rho c into R2 square divided by the diffusivity multiplied by the corresponding surface concentration. Remember that the experiment was conducted exactly under the same conditions. So, therefore, the concentration of the species at the surface of the reactor is same for both R1 and R2. So, that is equal to 3 times phi 12 cot hyperbolic phi 12 minus 1. So, now taking the ratio of these two expressions here, we can now find a relationship between the observed reaction rates for both runs and the corresponding Thiele modulus. So, by taking the ratio, we find that RA2 into R2 square divided by 1 into R1 square. So, that should be equal to phi 12 cot hyperbolic minus 1 divided by minus 1. So, this relates the information that has been observed experimentally and the corresponding Thiele moduli. So, the left hand side of this expression is essentially the one which actually contains all the information that have been estimated experimentally and the parameters or the properties of the system that is being used, and the right hand side is basically the Thiele moduli, ratio of the Thiele moduli, ratio of the function of Thiele moduli. So, now let us look at the expression for the Thiele modulus. So, phi 1 1, which is the Thiele modulus for the first order reaction for the first experiment, that is given by R 1, which is the radius of the pellet multiplied by square root of minus R A S, which is the reaction as if it were conducted on the surface concentration, multiplied by rho C divided by the diffusivity into the concentration of the species at the surface. And similarly, phi 1 2 will be R 2 into square root of minus R A S into rho C divided by d E into C A S. Now, from here, taking the ratio, dividing these two equations, we will find that phi 1 1 by phi 1 2, that should be equal to R 1 by R 2. So, the ratio of the Thiele modulus is given by the ratio of the radius of the pellet itself, and from here one can find out the relationship between the Thiele modulus under different experimental conditions. So, that will be phi 1 2 into R 1 by R 2. So, now plugging in the numbers, we will see that phi 1 1 is equal to 0.01 meter by 0. meter into phi 1 2. So, that is equal to 10 times the phi 1 2. So, the Thiele modulus under one experimental condition is about 10 times the Thiele modulus of the second experimental condition. So, that is the relationship that we get from the Thiele modulus of these two experimental conditions. So, now earlier we derived the relationship between the ratio of the reaction rates with the corresponding Thiele modulus. So, now we can plug in the relationship between the Thiele modulus under these two experimental conditions into that expression, and that will be minus R A 2 into R 2 square divided by minus R A 1 into R 1 square, and that should be equal to phi 1 2, which is the Thiele modulus for the second experimental run, into cot hyperbolic phi 1 2 minus 1 divided by 10 times phi 1 2 into cot hyperbolic 10 times phi 1 2 minus 1. So, all the information here is known, the only variable is only unknown is phi 1 2. So, we need to estimate phi 1 2 using all the other information that we already know. So, from here we can plug in the numbers. So, the reaction rate for the second condition will be 0.9 into 10 power minus 2 multiplied by the square of the corresponding radius divided by 0.18 into 10 power minus 2 into 0.01 the whole square, and that is equal to 0.05, and that will be equal to phi 1 2 cot hyperbolic phi 1 2 minus 1 divided by 10 times phi 1 2 into cot hyperbolic 10 times phi 1 2 minus 1. So, now by solving this expression, we can find out what is the Thiele modulus for the second experimental run. So, solving we can find that phi 1 2 is about 1.65. This is for R 2 equal to 1 millimeter. We now know we also know the relationship between the Thiele modulus of these two experimental conditions. So, from there we can find out phi 1 1, which is 10 times phi 1 2, that is equal to 16.5 for R 1 equal to 10 millimeters. So, we have now found the Thiele modulus. Now, once we know the Thiele modulus, we can actually estimate what is the effectiveness factor, because effectiveness factor and the Thiele modulus under given experimental conditions are actually related. So, for R 2, which is the second experimental run, eta 2 equal to 3 times phi 1 2 into cot hyperbolic phi 1 2 minus 1 divided by phi 1 2 square. And so, once we plug in the Thiele modulus data, so we will find out that this effectiveness factor is about 0.856. And then similarly for the first experimental run, the effectiveness factor eta 1 will be 0.182. Now, the next question is, can we find out what is the smallest radius at which the internal diffusion limitations does not exist? That is, there is no internal diffusion limitation that is limiting the reaction. So, suppose if we assume that the effectiveness factor should be 0.95, suppose if the effectiveness factor should be 0.95 at which there is no internal diffusion limitations, then we can actually use the relationship between the effectiveness factor and the Thiele modulus in order to find out what is the corresponding Thiele modulus. So, we know that 0.95, that is equal to 3 times 513 cot hyperbolic 513 minus 1 divided by 513 square. So, by solving this we can find out that 513 is about 0.9. So, that is the Thiele modulus, that should be the Thiele modulus if the reaction has to be conducted under no internal diffusional limitations. So, once we know the Thiele modulus, we also know what is the relationship between the Thiele modulus ratio and the corresponding radii. So, we can use that expression in order to find out what should be the particle radius in order for the internal diffusion limitations to not exist. So, 513 divided by 511. So, this is 511 is the Thiele modulus corresponding to one of the already conducted experiments, and so that will be R 3 by R 1. So, from here we can find out that R 3 should be equal to about 0.55 millimeters. The smallest pellet at which the internal diffusion will not limit the reaction is about 0.55 for the given set of experimental conditions. So, in this way, using some trial experiments, one can actually find out what should be the pellet size under which the reaction has to be conducted in order for the internal diffusion to not be present and not affect the reaction severely.